Yeah, like Leslie said, I have a PhD in microbial ecology and soil microbiology, so that's where my background really is. But I got involved in all kinds of areas of microbiology, microbial physiology, and molecular genetics, including synthetic biology. And so I have a very broad background and very broad interests. And David and I got together a while ago, and we looked at the situation of petroleum and fossil fuels and global climate warming and global climate change. And what does biology play? Can biology play a significant role in that? People have been looking at this long before we had, but we thought, is there anything new that we might be able to add to the mix? And is there something novel that we might be able to do that would make it more successful? So we thought about many things and decided upon the technology that I'm going to describe to you today, along with our marketing and a bit of our business strategy. And we formed a company called HelioBioSys. Today, I'm going to talk about bioproduced feedstock sugars for bioproducts. I'm not going to specifically talk about anything in particular and not about fuels, because fuels, biofuels, as many of you may know, really have been the focus of several companies that were developed in the course of the last 10 years. And one by one, they have all essentially reached the same endpoint in that the biofuel market is extraordinarily large, or I should say the fuel market is extraordinarily large and extraordinarily difficult to get into, and the practicality of doing that is very difficult. So, so a little bit about our company. We actually started with another nonprofit who sort of sponsored us in the very beginning, the SETI Institute, and we looked for screening cyanobacteria to produce ethanol. First idea was, well, can we just go straight to ethanol or some kind of bioproduct like that? And so we actually were successful in genetically modifying cyanobacteria to actually produce ethanol. What happened? Well, ethanol is toxic. They killed themselves. So we said, okay, what else can we do to modify it? So we decided that we would actually modify it a little bit, and we controlled the amount of ethanol production. And then the next problem was, okay, well, they are going to produce the ethanol. Ethanol is highly volatile, and we lost our product in the air. I might say that concomitant with this, there's another company that actually had the same idea called Alganol. However, Alganol decided they were not going to switch gears. They actually went all the way through. They produce ethanol. They had this, if you read their papers and read all the information over the years about that company, they had the same exact problems we have. And so they've developed a system now where they grow the organisms in closed bioreactors, constantly regulating ethanol production and harvesting the ethanol. Okay? They've been at it a long time. Then a few years later, we decided we were going to leave the SETI Institute. We incorporated ourselves and set up a self-funded lab, and we developed our cyanobacterial consortium that I'll talk to you about in the near future. I mean, within a few minutes. So then we started to characterize our organisms into consortia and started to actually try to do some scale up. And realizing that we're a small company and just two individuals, we had to really expand and develop some partnerships. So the first partnership we developed was with SRI International, and we're still working together. And then we expanded, and we're now working with the Lawrence Berkeley Lab division of the ABPDU, which stands for Advanced Biofuel Product Development Unit. We also have a partnership with Moss Landing Marine Lab, which is part of the Cal State system, and a private aquaculture company called Monterey Bluewater Farms. And just last year, we were successful in getting a small DOE grant with the ABPDU, and we're continuing to write proposals for outdoor scale-up and sugar characterization. And I'll get to one of the questions in a minute here towards the end of the lecture on what our strategy is for getting money in addition to federal funds and what kind of problems we have encountered when trying to get private funding through venture capitalists. So our future is to build partnerships and to commercialize. So basically, we rely on SRI International and Lawrence Berkeley National Labs for their laboratories because we can't really afford, as a small startup, to have much in the way of a laboratory. For actually scaling up to small-scale ponds, which is what we're trying to use, managed pond systems, up to a few thousand gallons, we've teamed up with Moss Landing Marine Lab. And then to go bigger than that, to 1 acre pond farms, we're teaming up with Monterey Bluewater farms. And of course, then, as our technology is basically a marine-based technology, we have to have a method for actually separating out salt from our product, and we're working with a company called Novacep and a company called Dow, and they both are involved in doing salt separation from sugars on a commercial scale. And then we also, as was mentioned just previously, we're also doing a techno-economic analysis, TEA, also with SRI, Berkeley National Labs, and Life Cycle Associates, which is a private company, as well as NREL. And a TEA basically is based on one of the things that Leslie mentioned earlier, and that is the whole analysis of the life cycle of your system. And I have an example of that. So one of the things that we have done that most other companies have not done is we've keyed in on cyanobacteria. So let me tell you a little bit about cyanobacteria. Cyanobacteria have been around a long time, so they're very hardy. They were the first oxygen-producing photosynthesizers to evolve. Our entire atmosphere actually came into being with oxygen because of them. They are distributed everywhere in the oceans, lakes, rivers, deserts. You find them everywhere. They're both marine and fresh water. They are robust. That's one of the reasons why we chose to work with them. They're prolific. They like to grow. They grow everywhere. And they're very well adapted to changes in the environment. And they'll grow everywhere from the poles to the equator. You find them in desert crusts. You find them in the ocean. And you find them in lakes and rivers. And one of the things that we really like about working with this organism that we found interesting and why we chose marine organisms is because marine cyanobacteria excrete sugars into the water. They don't just hold them inside the cell. They excrete the sugars into the water while they're growing, which really alleviates a lot of problems associated with getting anything out of a cell. And they have the highest photosynthetic efficiency of all organisms known. Now, this is just to illustrate here, and I don't have a pointer, but if you look at this, cyanobacteria evolved a long, long time ago, several billion years ago. And if you look at it, you see that as they grew and grew, see that line? That's the blue line in the very top in A there. Well, they start producing oxygen, and as they grew and they took over, they really produced a lot of oxygen, and it changed the entire as you might call it, the entire ecosystem of the Earth at that time. And it was because of these little cyanobacteria and their being so prolific and so photosynthetic efficient that it allowed the rest of the whole animal kingdom and plant kingdom to grow and develop to what we see and evolve to what we see today. In addition to that, cyanobacteria are really very important in photosynthesis that we see around today because they actually not only evolved the first photosynthetic apparatus. What happened then is there was something called endosymbiosis. And by endosymbiosis, what I mean is that there were other organisms that took them up. And when they took them up, they actually then became part of that organism. So in other words, the cyanobacterium was taken up by a eukaryotic organism. It carried out photosynthesis. That eukaryotic organism really got its energy and some of its sugar from that organism that it took up, that ancient cyanobacterium. And now it it lives now in symbiosis with them. And eventually what happened is that the organism that became symbiotic with the larger organism became so much a part of it that they integrated together, and that cyanobacterium became what is now known as a chloroplast for larger plants. So all oxygenic photosynthesis essentially arose from the cyanobacteria. So whether you're a tree or grass, you have the same photosynthetic apparatus as a cyanobacteria. Now, the other thing is that when David and I first went out into the world and talked about our idea of cyanobacteria, everybody just assumed that we were talking about little green slime, which we were, and that little green slime is, they all say it's algae. Well, it isn't. Cyanobacteria are very different than algae. Unfortunately, in the early days, cyanobacteria were called blue-green algae. And they're called blue-green because they look blue-green, but they're small and they're not algae. They're in a whole different domain. They're bacteria and not eukaryotic. So I put up this little table here that essentially gives an organismal comparison. One's a prokaryote. That means that the cyanobacteria does not have a nucleus, as do algae. They are bacteria. They're not, algae are not plants, but they are a whole different eukaryotic system unto themselves. The cyanobacteria are really small, whereas algae are large. Cyanobacteria tend to be unicellular, or they grow in filaments that you can see in a lot of places. Algae can be uni or multicellular. They both carry out photosynthesis. They both fix CO2. That means that they are autotrophic. They don't need any organic carbon. One of the big advantages for the type of work that we do with cyanobacteria is that they fix nitrogen. That means you don't have to fertilize. They get their nitrogen out of the air. They can take N2 and transform it into ammonia and use that. Okay, whereas algae need to be fertilized. Cyanobacteria are green, usually blue-green. Algae can be green, red, or brown, and they're quite different. And their habitats actually tend to be the same. They can live in the same areas. Algae are a little bit more finicky. They're a little bit more particular. They're not as diverse as the cyanobacteria. And cyanobacteria tend to reproduce vegetatively, whereas algae can reproduce sexually as well as vegetatively. Now, if we look at some of the process comparisons that I have up here, you can see that usually when people are working with organisms, they use them in a monoculture. And a lot of the biofuels and bioproducts that algae are used to make are used by genetically modified organisms. We, in our technology, do not use genetically modified organisms at all, and I'll explain why in a minute. Now, we produce sugars. Algae don't really produce sugars. Algae produce lipids, and those lipids can be used as a substitute for some petroleum-based things. In addition, algae have a different kind of cell wall. So they have a plant-like cell, more like a plant cell wall, where you can actually harvest the biomass and get sugars from the biomass, because they have sugars in their cell wall. And cyanobacteria do not. And I threw this up here strictly because, again, we get a lot of questions about cyanobacteria biomass. Why aren't you taking your biomass? What do you do with the biomass? We go into people and we talk to them and say, well, we make sugar, and the sugar goes out into the solution. And they say, well, great, what about the biomass? And well, we have to explain to them that cyanobacteria are different. They don't have sugars in their cell wall. What they have is they have what's called, and I threw this up here just to illustrate it, they have what's called a peptidoglycan, which is not cellulose, not sugars. It's made up of this other stuff that you see on the right side. What it is they're essentially peptides that link together N-acetylmuramic acid and N-acetylglucosamine, which are sugar-like, but they're not sugars. Okay? They are amino acids, sugars. Far cry from cellulose. So they're really quite different. So I want to get the point across that biomass is not really that important to us as far as our product is concerned. What is important, and one of the things that we do, is we use ecomimicry. You may have heard of biomimicry, which is essentially, and biomimetics, which is an imitation of the model systems and elements of nature to solve complex problems. But what we do is ecomimicry. So what we do is we try to really just mimic how a natural ecosystem functions. And that is really what we wanted to do. That's why we come up, one of the reasons why we came up with this whole idea of a consortium rather than a monoculture growing just one organism. Okay? So what it really is putting together our own type of ecosystem that mimics nature. Okay? There we go. So what is our technology? Basically, we have a very I won't say limited, but a very small supply chain. All we really need is seawater, sunlight, and air in our organisms. And that's it. And from that, we produce sugars. And I said earlier that the organisms that we have are the most photosynthetic, that in general, Cyanobacteria are the most photosynthetically efficient organism there is, and they are. And we've put together a consortium of these cyanobacteria that have the characteristics of growing quickly, they have a short generation time, and they're very efficient at producing and excreting sugars. So here you see this little table. It may not mean much, but essentially I can say that we go from what's called the actual sunlight that organisms use, which is called PAR, photosynthetically active radiation, which is the spectrum between 400 and 700 nanometers. And they can produce sugars at about over the percent level, much over the percent level, whereas most other organisms had a total, when I say most, I mean like trees and grasses, including switchgrass and everything, has a photosynthetic efficiency an order of magnitude less, total photosynthetic efficiency an order of magnitude less. That's the other reason why we chose these organisms, is because they're energy efficient. So basically, to summarize, we take sunlight, carbon dioxide, nitrogen from the air, and seawater, which has all the micronutrients and everything else in it. That goes into the cyanobacteria. The cyanobacteria produce fermentable sugars and polysaccharides. And we also have the leftover cell of the cyanobacteria. We call that biomass, but we use it for an entirely different reason than other technologies do. Okay. So let's get a little bit into the whole business aspect of this. Now, sugar as a commodity is used in a lot of places, industrially as well as for food. So this is raw sugar price over the course of several years. So the sugar price has gone up, and this ends in about 2012. And the sugar demand has gone up. I will say that the sugar demand has continued to grow, but the sugar price last year started to plummet. It's now down to 11 cents. Now you may say, why did that happen? Well, and it's going to go back up. Everybody predicts it's going to go back up to over 20 cents a pound. The reason it fell is because you have to pay attention to international politics. What happened was sugarcane in Brazil, the price fell. The price fell in sugarcane in Brazil because the whole economy in Brazil was going through a change, a negative change, I might add. And so the cost of their money was not worth as much. Since their money was not worth as much, the cost of the sugar cane on the international market dropped as well. So that had a dramatic effect, and they're big producers of sugar. That had a dramatic effect on the cost of sugar, and that's why it went down. But it's It's projected to go back up. This green line you see at the bottom, that is our projected price that we can sell it for. Not a projected cost, but a projected price. When I say cost, that means cost of production. So our projected price is we can go 7 to 10 cents a pound easily. If the price goes up, there's no reason why we couldn't raise our price up as well and make a larger profit. So our whole business premise is to produce low-priced sugar for the sugar market and our customers. And that means high customer margins. That means a greater profit margin for them in a greater than $30 billion market in the United States alone. Okay, so if we take a look at what we have our cyanobacteria. Okay, we then grow the cyanobacteria. We separate the cyanobacteria cells from the sugar solution. So we have a sugar solution. We desalinate the sugar solution. And if you follow the cyanobacteria sugar feedstock, we have a polysaccharide that can go directly to producing things such as plastics or other polymers. And by the way, I will say that we have recently been successful in taking our polysaccharide produced by our cyanobacteria and making plastic from it on the lab scale, but we have demonstrated that that can be done. And then we take the mono, di, and trisaccharides, and we've shown that they can be used by other organisms to grow and to produce other bioproducts. So in other words, fermentation. We've shown we can produce ethanol, butanol, and other bioproducts. Then we can take the residual biomass and it can be used as fish food or a fish food supplement. It can be used as a fertilizer, and it can be used in anaerobic digesters to produce compost or power. Now, let's look at some productivity comparisons between cyanobacterial produced sugars and sugarcane and corn. Now, cyanobacteria, I said, grow really, really quickly, and they do. And they produce a fair amount of sugars. So basically, we have done, granted, we haven't done it all the way to an acre, but we have shown on the basis of larger than laboratory scale, I will say that, and not in the confines of a clean room, outside in the real world, we can show that we should be able to produce 100 tons of sugar per acre per year. Now compare that to sugarcane and corn sugar. They produce on the order of 5 tons of sugar per acre per year. So even if our 100 tons of sugar per acre per year is way off by 50%, and we only produce 50 tons, we're still an order of magnitude better than they are. Now, one of the things that we always get asked is, so what about the energy density of sugar? And what can you really do with sugar? And how does it compare to other things? Well, that's why I threw this up here. If you look at the energy density, which is actually the joules per gram or megajoules per kilogram, you see that wood down there is on the order of 8. Ethanol is on the order of 25. Coal is on the order of 30, oil is 40, and gas is way up there about 52 or 53. Well, sugar is 17. Plain old glucose is 17. So plain old glucose is right above wood and below ethanol. So if you're just going to use sugar, that gives you an energy density comparison. Now, what about productivity comparison? So if we look here at productivity comparison, and here, okay, we're making sugar. The world doesn't necessarily always like to run on sugar because it hasn't in the past. So if we try to compare it in looking at ethanol per acre per year, gallons of ethanol per acre per year, and you can actually do that calculation because it takes about 13 pounds of sugar to make a gallon of ethanol. If we look at corn, is way down here at about 2. Sugar cane is about 3.5. Machanthus, or not 3.5, I'm sorry, this is 1,000, sorry. Okay, Machanthus comes next. Algae is way up there, But cyanobacteria is way, way up there, and that's also because they grow so quickly and so photosynthetically efficient. They're 8 times the productivity of miscanthus. So we were fortunate enough to be selected. We applied for and we were selected by NREL, the National Renewable Energy Lab, in their NCAP, what they call National Commercialization Assistance Program, to develop a techno-economic model, which looks at the production and purification of our cyanobacterial sugars, to estimate the key mass and energy balances, and to quantify associated equipment, capital, and operating costs. That means centrifuges, piping, pumps, desalination. Do you need to add CO2? How much mixing? How much energy is it going to take for the whole system? And then calculate a minimum sugar price. This system also has, it calculates as part of it, biomass and production ponds. And when I mean propagation and production ponds, what we do and what is done in the industry for algae and everything else that you're trying, organisms like that that you're trying to grow at large scale, is you start out small. And then you go like from a liter to 5 liters, and then to 25 liters, then to 250, because you can't go from a liter to 100 liters. Because what happens is you have, your population is so dilute that it usually doesn't, it's not healthy. It takes too long for it to really take off and grow. So you have to make sure you have at least a minimum amount of organisms that you go from one stage to the next. In addition to that, speaking of propagation, you really want to make your organisms grow quickly and outgrow anything else that might be in there. When you're talking about large-scale operations, commercial-scale operations, you can keep them clean, but it's very hard to keep them sterile. It can be done, as is done in the pharmaceutical industry. But if you look at the price of some of the pharmaceutical products versus the price of what you might want in a commodity market for biofuels or bioproducts, there's a huge difference. So you pay a huge cost for keeping everything sterile. So you want to keep it clean as possible, but it's usually not sterile. So you want to have the organisms in there, in your system, as many of the ones that you want in there, and contaminants as low as possible, and you want your organisms to grow faster and outgrow any contaminants that are in there to make the product you want. So that's why when you propagate in larger quantities until you get to your production pond. Yes?
Isn't that the same challenge you face in brewing beer?
Well, beer production, yes, sort of. Actually, if you go through beer, they propagate as well. It's propagated from a small amount to a large amount to a large amount, but they're sterile. They're fairly sterile. Okay? They have an enclosed system. If you ever go through a brewery, you'll see that it's fairly close. But again, the cost of a beer is a lot more than the cost of what you want for these bioproducts. Yes. If you get contaminated beer, it usually turns to vinegar, malt vinegar, and that's not what you want. But yes, so yes, you're absolutely right. There they start with an enclosed system as well. So this model that NREL worked up for us also looks at centrifugation and solid separation to remove biomass, sugar recovery and purification operations, in other words, desalination primarily. Co-products and waste products assigned are financial value. By co-products, I'm talking about essentially taking some of the biomass, using it for fish food as well. Okay, now, as part of a techno-economic analysis, you do a life cycle analysis, as Leslie mentioned before. And she mentioned a greenhouse gas analysis. That's the GHG. So what we have done here is we actually had somebody who does this for a living, is a professional, roughly go through our system and look at it and see how it would compare to a petroleum-based system or gasoline. And so we don't really have any chemical or fertilizers that we add, so we're zero. We do have to cultivate the system, and so there is a greenhouse gas emission equivalent. That's what's on the x-axis there, I mean the y-axis. And so we have some cultivation greenhouse gas emission. We have separation desalination greenhouse gas emission. We have some, if we're going to a biofuel, we have the product or sugars of or product of any kind, we have a greenhouse gas emission. Transportation and delivery costs, and then we move up to combustion. If this was based on if we were making a biofuel, combustion. And so we have a total of about 45 greenhouse gas emission equivalents. Whereas you look at petroleum gasoline, you have about 95. So in this rough estimate calculation of greenhouse gas emissions, cyanobacteria actually are pretty good. And one of the reasons why they're really good is because this first thing that says chemicals and fertilizers, we don't need any. Most other things do. And the cost of producing fertilizers, especially nitrogen fertilizers, is very, very energy intensive. So I threw this in here because one of the questions that I got was market comparisons and competitors. Who do we have as competitors? Well, our main competitors and the people who would be our customers now buy sugar from companies like ADM, Cargill, and Bunge. And so what about other companies that are sort of in the market that produce things like sorghum? Well, there's a company called Chromatin that will sell you the plants for sorghum, and they'll sell it to you for about 10 cents a pound. And then what about cellulosic? Okay, there are companies that are entering the commercial phase, that are actually in the commercial phase. I've listed some of them here. And breaking down cellulose is not an easy, inexpensive thing to do. Okay? So there are companies like Renomatics that uses supercritical water. Other companies use acids. So it's very difficult to break down cellulose, and it tends to be expensive. But it's happening. The only company that's out there that really does something similar to us is a company called Protero. And they're an early stage company, and they use a cyanobacteria that is genetically modified to excrete sucrose. And it's a freshwater cyanobacteria. So what they have done is they have made it a very efficient sucrose-producing organism, and they have genetically modified it so that they have in the membrane, a sucrose membrane transporter that whenever the sucrose is made inside the cell, it transports outside the cell. Okay? Now, I should say that they originally started in biofuels, open ponds. They have progressed to where they are now, making nutraceuticals and other things that sucrose could be used for that's on a smaller scale, and they use an enclosed bioreactor. So if we want to look at our company and how do we project our technology development from the prototype to the pilot plant to the commercial stage, well, basically we've already got in place combining ourselves with and partnering with Moss Landing Marine Lab, SRI, and the ABPDU unit of the Lawrence Berkeley Labs to start a small-scale scale-up to verify productivity, doing some things outside at the thousands of gallons level, and to do sugar analysis as well as stability of our population. The next stage, of course, is to go to the pilot stage, and there we target 1 acre. We target 1 acre as our commercial-sized pond. So instead of having huge multi-acre ponds, what we want is 1 acre pond that's duplicated many, many times. What we call a pond farm, a full-scale commercial pond farm, and what we based our financial calculations on, is essentially we call it a 5,000 acre pond farm. That really consists of about 4,600 to 4,800 acres of actual pond, including the propagation pond, and then the connecting roads and field laboratory and analyses area to go along with it. So that's that would be a commercial scale. So we want to go, we're really starting out with a few thousand gallons, go to our pilot, which is a 1 acre, test it out at 1 acre, then commercial is to multiply that 1 acre. The other thing that this really hinges upon, as do all other large-scale sugar production for industrial purposes hinge on, is that there's co-location. So your customers have to be co-located with you. That is not anything new to us. It's the industry standard. For example, Solazyme, which is not a large company, but they're around in South San Francisco. Their actual production facility is not here. Their production facility is in Iowa. Why? Because they buy their sugar from ADM, which is corn. And so they're next to a corn sugar production plant. They're co-located. Other companies do the same thing. So we would have to do the same thing because it's just too expensive to transport these things all across the nation. So this is our big picture of an integrated coastal biorefinery where we have our cyanobacteria production. We then go to making our sugars, which go into various products by our partners. We actually then would partner with an aquaculture for actually purifying water and for producing fish food. I showed part of this to you earlier. But that's our big picture of an integrated coastal biorefinery. When you get into this kind of a system, this also feeds into the techno-economic analysis so that you don't have much in the way of waste products. You recycle as much of the waste products as you possibly can. Okay. So that really is the end of the formal part of the lecture. However, Leslie sent me several questions that I think are really good questions. And I'll answer some of them now. So I'll answer all the questions he sent me, and then you can ask more. So someone asked, what are the potential environmental or biohazards of artificially growing the cyanobacteria? And I just wrote quick and dirty answers there to remind myself. There really is none. And the reason for that is because these organisms are naturally occurring. So you find them blowing around on the coast, in the coastal waters everywhere anyway. The only thing different we have done is we've selected those organisms that can grow together and do grow together and grow together more efficiently, synergistically, to produce sugars. Now, so there's nothing really strange about this. The only thing that we've done is patented the way that we've put them together and to allow them to grow together. That's all. So they're out there anyway. So there's no environmental biohazard as far as contamination really is concerned. Someone asked, can the method that we use be modified to desalinate salt water? The answer to that, yes, because the methods that are used are really desalination methods that have been adapted for us to use to take away the sugar from the salt. So there's nothing new in that particular technology there at all. It's just standard desalination type of technology. We have chosen to initially test microfiltration, and there's nothing new about that either. Another question, between developing this new technique for creating sugar and finding investors for your company, what was the most difficult part you found in starting HelioBioSys? Well, one thing, when we were talking to investors, we had a very difficult time convincing them that cyanobacteria are not algae, and that the problems that we have with cyanobacteria are different than the problems that you have with algae. That, for some reason, when we first got involved in this, we did not expect that. But it has been a problem and a difficulty that we know we have to face, so we try to explain it as best we can as we go along. We also try to explain that, as I said earlier, biomass. Everybody keys in on biomass. Well, cyanobacterial biomass is quite different than algal biomass or plant biomass. In fact, the DOE has specific calls out there for to try to take biomass, as they call it biomass, algae, trees, switchgrass, and convert that to sugars. Okay? We can't do that because, as I showed you, our biomass doesn't have regular sugars in it. And so trying to make people understand that has also been something that we've had to educate ourselves on how to educate other people about. And the other big thing is de-risking. People look, or I should say VCs look at a business, and one of the things they try to see is, what are all the risks? And what are all the big hurdles? And have you passed them yet? And because there's been so much work that has been done on algae and trying to produce large quantities of algae to produce biofuels or biomass in open ponds, it's been extraordinarily difficult for them to do that. So they ask us to, what is your scale up? So scaling up is one of the biggest risks that we have. The thing that we have that I did not mention as far as scale up is concerned is that when we use a consortium of cyanobacteria, ecomimicry is key. And part of that means that they are resistant to contamination and to predation and to being taken over by other things coming around. Okay, why is that? It's because it's a community, and the community is more resistant, inherently more resistant. One of the reasons that they make a polysaccharide, by the way, is so that they engulf this community in polysaccharide. And if you look at it under a microscope, you can see predators trying to go after the cyanobacteria and eat them, but you can't. They can't because they can't get through the polysaccharide. So that's one reason why we've used this kind of technology, is to really protect our organisms from the natural environment in the way that, that organisms growing in the wild protect themselves as well. Another question, as far as cyanobacterially produced sugars goes, this is certainly a novel concept. Is there yes, it is. Is there a market for this kind of product? Yes. Sugar basically is sugar. Polysaccharides are polysaccharides. Doesn't matter who makes them, whether they be made by cyanobacteria or whether they be made by breaking down a tree, lignocellulose, or cane sugar or corn sugar. Sugar is sugar. The thing that we do that's different is we make a wider variety of sugars, not just one kind of sugar. But the sugar is not novel in and of itself. And who are your competitors? Well, I listed them earlier. Things like companies as big as ADM and Cargill to small companies such as Protero. Okay, what is the scalability of this product? Potential revenue of this kind of work, 1 acre pond. Well, a 1 acre pond is our basic unit. And we figure that we can, using our 1 acre pond as our basic unit, one of the two most costly things to do with this technology, one has to do with construction, and that is a pond liner. Believe it or not, 1 acre pond liner would cost on the order of $19,000 to $20,000. The other cost that really drives our production up is desalination. Desalination, of that approximately 7 to 8, 9 cents a pound, desalination accounts for a good portion of that. What grows good for the environment? Well, we do not compete with anything that has to do with food, and we use non-arable land. So those are just two things. The process that we use doesn't use fertilizers. We don't have to fertilize. We don't have to use any pesticides or anything like that. So in that regard, it's non-toxic and non-contaminating. Did your idea come out of the Asimov yeast farms? No, it didn't, actually. It did not. It just came out of us sitting around talking and figure, how can we most efficiently produce sugar? Your website says that the bacteria you use are non-GMO. Why does this matter? I didn't say anything earlier because I knew this question was coming up. Well, genetically modified organisms means that you're taking them and you're making them do something they don't want to do. So, for example, if you genetically modify an organism to produce a substance, another organic substance, and excrete it, I don't care whether it's an extra sugar or a carbohydrate or a lipid or whatever, it's excreting it. That means it's not using that for itself. It means it's expending energy to make it and excrete it. So it's not doing it any good. So it doesn't like to do that. So those organisms that are genetically modified tend to not grow quite as well. The other thing that tends to happen is, especially when you start to scale up, it's very difficult to control the population for a longer period of time when you scale up. What happens is, because these organisms don't like to do what you're forcing them to do, you have to put a selective pressure on the population. One of the most common ways to do that is to take an antibiotic-resistant gene, hook it to the gene that you're interested in to make your product, and then put that in the organism, and then put that out there, and then grow them in the presence of the antibiotic so that the wild type doesn't grow. The only thing that grows are your organisms. Well, you can imagine that doing that on a large scale gets very difficult. And what happens is that the organism that you've genetically modified fights what you're trying to do, and if it can mutate back to the wild type, it will, and it does. So from a technical standpoint, using genetically modified organisms in systems that you're not going to tightly control because it costs so much money is very difficult to do. In addition to that, if you're out there and want to run like a managed or even semi-managed open pond or even a large type bioreactor, and you're using genetically modified organisms, you've got to go through a lot of regulatory issues. You have a lot of regulatory issues to deal with. So you have to comply with all the regulations that are out there to not let your GMO out, so that it doesn't get out in the wild. Can't genetic modification be helpful producing materials that are useful with better yield and lower costs? Yes. And in fact, it is done. And if you do it right, it can be very productive and very good. But in certain circumstances, it's not economically efficient, or the regulations are too hard to comply with. So we found that for us and what we want to do, we didn't need any genetic modification, so we don't do it. And I think that's the last question. Are humans or animals consuming a lot of what you produce? No. Our sugars are not produced for human or animal consumption. They're produced for yeast and other bacteria to eat. Why should we be worried about the effects of the GMOs? And I think I alluded to that earlier, and that it's not a health effect necessarily that we're talking about. We're talking about a technical effect of their being so difficult to manage, regulatory issues. And that's basically the reason why we want to stay away from GMOs. Now, are there any other questions? Yes.
Why can't humans consume your sugar?
Why can't humans consume the sugars? Well, you could if you purified it. We don't have to go through such a high rate of purification if we're just feeding other bacteria or yeast. It's just a purification. And the only reason why you don't do that is because it costs money. So we want to produce the sugars at the very lowest cost of production for industrial use. Yeah, glucose is glucose. Sucrose is sucrose. Yes?
Another question. So in the life cycle analysis, the desalination was the most carbon intensive part of the process?
Yeah, because it takes a lot of yes, because it takes a lot of energy.
Yeah, have you guys been researching ways to reduce
yes, and the whole desalination aspect is probably one of the, I won't say riskiest, but it is one of the areas that we really have to balance cost versus energy output, because there are techniques out there that would work really well and not be so not produce greenhouse gases, but they're very expensive. So it becomes a matter of balancing the expense versus the greenhouse gas production. I mean, and energy intensiveness. one of the problems, as you may or may not know, is that for the smaller sugars, for the mono, di, and trisaccharides, the size of those on a grand scale is not that much different than the salts. So it makes it more technically difficult. Any other questions? Yes.
How sensitive are the ponds to changes in weather conditions?
Oh, it's not the ponds necessarily. You're talking about the organisms. That was another important factor in why we chose these organisms that we did. Okay. So what happens if you have a pond, it doesn't even have to be a big one, and you put it out there and it's an unmanaged, just open pond, several things happen. One is that all kinds of debris falls in. Another one is that the water evaporates, changing the salt concentration. So what you need then is and that's why we say we use managed ponds, which means that we put sort of like a screening on top of it to keep so much debris from falling in. The other thing that does is it helps control the evaporation. So, but what if you can't control the evaporation? And in your pond, and what if you have a sudden rainstorm? Unlikely as that it may be here presently, but what if you do? You change the salinity drastically. So what happens? Well, if you don't have the right kind of organisms and you change the salinity, they die. Both if it gets too dilute and when it gets too saline. So we have chosen a group of organisms that will withstand a large environmental change in salinity, essentially take them down to physiological saline, and they won't like it, but they don't die, and then put them back up to seawater, which is about 3.5%, and they're fine. Now, if you increase the salinity too far, like if you get it to saturated salt, they won't like that either, and they can die too. The other thing you have to look at is and we've looked at this as well, and that is temperature ranges. What happens if the temperature gets too cold? These organisms don't really care if it gets too cold. They do care if it gets too hot. If you actually raise the temperature to above 55 degrees C, they're dead. That's all there is to it. I will say that there are cyanobacteria that will grow up to 72 degrees C. But once you get past 72 degrees C, no cyanobacteria grows, simply because at 72 degrees C, you get hotter than that, chlorophyll falls apart. So that's the temperature limitation there. But these we think are pretty good for what we want. And then you have to look at pH range changes, and they also have a wide tolerance for pH too. Yes. Yes.
Can I just follow up?
Sure.
So also the amount of water that you let evaporate, I would assume means you wouldn't necessarily want to do it in a place. I mean, I know that you rely on sunlight for fuel, but then if you have a large water resource requirement too, then something like California.
The water comes from the ocean. So anything that we've talked about has to be coastal. So all you do is you have a pipe that goes into the ocean. And in fact, we use water out from Santa Cruz. We've used Moss Landing water. We've used, so what happens is there are stations out there that have pumps. And Moss Landing Marine Lab has one. Santa Cruz, UCSC has one out there. All they do is they have this big pipe that goes out in the ocean. They have all the permits done, and it goes through a sand filter, and that's the water that we've used. And that would be the same way if you have a larger system. Monterey, Bluewater Farms has the same thing, except on a larger scale, because they're growing fish in an aquaculture situation.
And there's no issue with the bacteria being in the dark for days at a time if it's really cloudy, because there's a sort of
Cloudy is not dark. These organisms are photosynthetically efficient, not just when the sun's out, but you can reduce the sun to 1%, and they'll still photosynthesize. They're not happy, but they'll photosynthesize. And they have to go through a dark cycle. So that's the other thing, whenever we grow these, we have two ways of growing them. One is in the lab, the other one's outside. The outside ones really like it, because they have a natural diurnal cycle. In the lab, you have to turn the lights on and off. When I say natural diurnal cycle, that means that the sun comes up, reaches a peak, and then it goes down. And that's how they've evolved. And so if you look at the actual physiology and metabolism of the organism during the course of the day, it's not the same. It changes, especially around noon to 2 o'clock in the afternoon. Noon to 2 o'clock in the afternoon, CO2 fixation goes way down. Then it starts to come up again. So they like that, and that has to do with changes in the luminosity and the solar flux. So they actually like being out there in the natural environment. So they change during the course of the day. Being cloudy on a day or two, that happens in nature. And that's really the way these guys like to live. Yes?
If desalination is such a huge part of the process, and I know that there was a company doing freshwater cyanobacteria, would doing a freshwater cyanobacteria with a lower yield offset that desalination?
No, cyanobacteria, freshwater cyanobacteria, you wouldn't have a lower yield, you'd have no yield unless you genetically modified it. Then you run into all the problems with a genetically modified organism. Yes.
You have a remarkable history of growing collaborations and, being able to co-locate with other operations where you've been really leveraged. What kind of guidelines or hints? Can you explain how you're so good at that?
I don't know, really. I mean, for example, when we approached, the first group that we approached was SRI. Okay, what we did is we approached them, and so basically their business and their science group, and we just gave them a presentation, a mostly technical presentation. Said, look, this is what we have, this is what we want to do, this is how we see you fitting in with us. How do you see fitting in with us? Would you like to be part of this, given that it's high risk? And they said yes, basically. So it's basically selling them on the technology. Then for Moss Landing Marine Lab, for example, they have a group down there that, and part of their charter is to work with small startup companies who are doing the kinds of things that we're doing. If that small startup company also will allow and has the capability to work with graduate students. So, when we talked with them, said, we think that this would be a great opportunity for a master's thesis on this particular part of it. And we're fine with that, and we can help guide that. And you have all the infrastructure and permits and everything to take water out of the ocean and put it back. And we have this technology on cyanobacteria and growing communities together. Is there something we can do? And the answer was yes. And when we approached Monterey Bluewater Farms, which is the aquaculture company, one of the problems that they really have is when they put that water back into the ocean, they have to make sure it's fairly clean. One of the big problems they have is with nitrogenous waste from the fish. Well, I said that our organisms fix nitrogen. They do. But they also have the ability to eat nitrogen out of the water if it's there. So what we propose to them is that our cyanobacteria, once we're through with them, we can put them in another tank or pond, and you can put your wastewater through it, and they'll take out all the nitrogen, and you can put it back into the ocean without all the nitrogen in it. In addition, some of that can be used as a fish food supplement. And they thought that would be a great idea to try, because it would save them a lot of money.
I'm hearing very systems-oriented view as you're looking at various people.
Well, we take the approach that you really have to be a systems-oriented if you're going to actually make a success out of it. If you're just going to do one small thing, it's probably not going to be successful. That's why, and I went over this quickly, but that's why we put together this plan for an integrated coastal biorefinery, is to get everything together to do as much as you can do with what you have. Because if you don't, you're going to end up, I think it's going to be very, very energy inefficient and cost a fair amount of money.
You haven't said lots about the bioplastics, but you've made demonstrations.
Yeah, that's reasonably. One of the things that we've done is we've looked at the whole idea of taking the polysaccharides and making bioplastics out of them. Bioplastics, come in two categories. They come in categories that are not so biodegradable, and they come in categories that are biodegradable, even though they're all bioplastics. And so what we wanted to be able to do was take our polysaccharide, which is biodegradable, convert that to a plastic, to see if we can convert that to a plastic, and see if there might be uses. How hard would that be, and how much would that cost? Well, we've teamed with the ABPDU. We have a grant from the DOE to have the ABPDU essentially do sugar analyses for us, do polysaccharide analyses, polysaccharide deconstruction, look at preliminary production of bioplastics, look at more details as far as using the sugars for various fermentation reactions. And so one of the things that they've done is they have taken our polysaccharide and have made bioplastic out of it. It seems to be a bioplastic that is not too brittle, which is good. So yes, we've taken the first step in showing that our polysaccharide, without really cleaning it, even doing too much to it, they added glycerin and did a mild acid hydrolysis and produced a bioplastic. It's just a blob, but it's plastic. Yes.
So you said that you have to be coastal, which makes sense, but coastal land can be very expensive. It's a prime vacation area, that sort of thing. And so, is that a major portion of your production costs, or?
We have taken that into account. We would not go to the coast of California. We would not use prime coastal land because you can't afford it. However, if you look at land across the world, there are coastal areas that are not that expensive.
But if you need to be co-located with a customer, then doesn't that put some more constraints on where
It does, because your customer then has to do some shipping. So it depends on who your customer is. But there are places within not that far distance. For example, if you were going to go across the border into Baja, there are ponds there, as a matter of fact. Okay, and so you just have to deal with international shipping. I also know that that Solazyme, for example, is in Brazil. There also Amyris is in Spain. So you can go. And there are companies in Singapore.
Could you alternatively have offshore artificial ponds?
That'd be very expensive. There's a group that was still looking at growing algae offshore in essentially closed bioreactor of bags. And if you go online and you look at Project Omega, you can find it. But it has some problems.
It's expensive just because you need to keep things sealed off from the ocean.
Well, it's expensive.
The infrastructure.
There are a lot of technical problems. Yes.
would the worst use of these sugars be to turn them into something similar to gasoline, like petroleum replacement?
The easiest, the sugars, what you do is you'd feed it to an organism, for example, that Amyris has. Okay, Amyris is a company in South San Francisco. They make something called farnesene. Okay, and from farnesene, you can produce a variety of biofuels. So you take your sugars and you feed it to you sell it to them, you feed it to their organism, they make the biofuels. But going directly, or you feed it to yeast to produce ethanol. So you have to feed it to someone, or you feed it to Clostridium to produce butanol. You have to give it to some other organism to produce actually the predecessor to the fuel or the fuel itself.
But in terms of efficiency, it would be where you would want to focus your efforts. You would rather be doing bioplastics or...
Well, no. Actually, where we focus our efforts is just producing the sugar.
So you don't want anything to do with the downstream?
No. Oh, the only thing that we do with regard to, for example, the bioplastics and the other fermentation things that we do is to show the efficiency at which it can be done and the process under which it can be done. And then go to other companies who actually do it and say, you should use our polysaccharide to make the plastic because it's more efficient or whatever.